Good morning. In this video, we are going to explain the characteristics of cancer cells in a simplified way and the effect of the inhibitory protein called P53 to protect or in protecting the body against cancer. But we cannot discuss the characteristics of cancer cells, which are completely different from or than the characteristics of normal cells without first reviewing what we have said in previous videos about the cell cycle, because they are closely related to each other. What is the cell cycle? The cell cycle is an ordered series of events that are repeated in an organized way in the cell. And during that ordered, organized series of events, the cell is allowed to grow in size, to make copies of its DNA before dividing into new daughter cells. And here we are focusing mainly about mitotic division of the cells. As you know from the previous discussion in previous videos of the cell cycle, we can divide those events that are regularly repeated in the life of a cell into two broad divisions, the interphase and the mitotic phase. The interphase is all about preparing the cell for mitotic division, and the mitotic phase is the phase where the nucleus will start dividing, or the DNA will start dividing, followed by division of the cytoplasm or cytokinesis. So we have karyokinesis, or the division of the nucleus and DNA, followed by cytokinesis, as we said before. The interphase, which is the preparation phase, consists of three phases, which are growth phase 1, as you know, S phase or synthesis phase, in which the DNA is multiplied, and the G2 phase or growth 2 phase that follows the S phase. And we're going to see all these phases on the following diagram, or on the diagram that will come very soon on the screen. But since that cell cycle, or every cell cycle, is an important event in the life of a cell, and since it has to be done in a very accurate way in order to prevent the emergence of abnormal cells or damaged cells, then there should be a strict follow-up from the part of the proteins and enzymes in the cell. These regulatory proteins and enzymes are called the checkpoints. So there is strict surveillance or regulatory control on the cell cycle, especially through proteins, whether they are enzymes or non-enzymatic proteins. And these checkpoints that are regulatory points or regulatory proteins actually will act at specific timings of the cell cycle, or at specific points on the diagram that represents the cell cycle. And the aim of the checkpoints, as I've just explained, is to make sure that only those cells that have intact DNA and in which the replication of DNA has been done not only accurately but also completely, meaning that all the chromosomes have been accurately replicated. Only those cells with intact copies of DNA and complete copies of DNA, for example, a new complete set of DNA to enable the cell to distribute this copied DNA on two daughter cells. But there is also a third condition that is very important, which is the favorable conditions, meaning that the external and internal conditions should be favorable for the process of replication. Needless to say that the purpose of these checkpoints, according to what we have read in the definition, is to prevent any cell that has a damaged DNA or an abnormal DNA from replication. And this is very important to prevent mutation, to prevent the emergence or the appearance of abnormal cells, because the abnormal cells will not only be abnormal in structure, but also abnormal in function, and this might result in serious damage or diseases affecting the living organism. And by preventing any cell with damaged DNA from replication, we are ensuring maintaining a stable genome, or we are ensuring that we are maintaining genomic stability. Because one important thing is that the genome stays constant over the generations to preserve the characteristics of the species. Here we can see a diagram that shows the phases of the cell cycle. So we have the G1 phase, the first growth phase, and then the phase of DNA synthesis and replication, and then we have the second growth phase, and then we have the M phase or mitotic phase. We have three important checkpoints, as we explained before. We have the cell growth checkpoint, G1 checkpoint, that is present at the end of the first growth phase, and it checks whether the cell is big enough, because during the G1 phase, the cell is accumulating nutrition to make sure that it has enough organic material to grow and to build new copies of DNA. So during this G1 checkpoint, the cell is checking whether its size is adequate, whether it has all the proper proteins that are needed for the next phase, which is the S phase. So what happens if during this check by the check proteins in the body or the checkpoints in the body, they discover that there isn't enough or there aren't enough molecules necessary for the synthesis phase, or if they discover that there are any abnormal proteins or DNA molecules, then what happens is that the cell will be forced to go into the G0 phase or G0 phase, which is a resting phase. Why is the cell triggered to stop? What do we mean by forcing the cell to enter the G0 phase? We mean that the cell is arrested, is forced to arrest. It's not allowed to continue and pursue its journey in the cell cycle. And this resting phase is an attempt from the part of the cell to try to repair the damage that is present in the cell, even if it is a nuclear damage or a nucleic acid damage. If the damage is repairable and if the enzymes in the cells succeed in repairing the damage, then the cell is allowed to re-enter the cycle, meaning that the whole process of synthesis of DNA and mitosis will be resumed. But if not, then the cell will stay permanently in the G0 phase and might undergo apoptosis later on. And we have discussed apoptosis in previous videos that you will find on the channel. On the other hand, we have another checkpoint, which is called the DNA synthesis point, and it occurs about the at about the middle of the S phase, or half the way long, and this checkpoint is checking whether the DNA replication has been done correctly or not. And this is very important, because if replication of DNA carries inaccuracies or errors, then the cell should not be allowed to continue, because if it continues, it will undergo division, nuclear division first and then cytoplasmic division, and it will give rise to abnormal daughter cells. So it should be prevented from continuing the cell cycle. And then number 3, we have the mitosis checkpoint, and the mitosis checkpoint occurs during the M phase, and it checks whether the spindle fibers, that's why we call it sometimes the spindle checkpoint. Here it's written on the diagram, it checks whether mitosis is complete. Actually, this is too broad and inaccurate. The checkpoint here checks that the spindle fibers that arise from the centrioles at the poles of the cell have been attached accurately to the centromeres of the chromosomes, and that the chromosomes are accurately located on the equator of the cell in metaphase. And if this is okay, then nuclear division is allowed, followed by cytoplasmic division. So as the cell is progressing across the cell cycle, every now and then the checkpoints or the regulatory proteins that are timed or that interfere at particular specific moments in through the cell cycle or during the cell cycle have to make the very important decision of whether the cell will continue and pursue or will stop and will be discarded. So what are the factors that affect the decision of these regulatory proteins, and therefore the decision of the whole cell to pause and go into the G0 phase or progress and continue in the cell cycle? The factors are many, but we are dividing them here into 2 broad categories. The first one is the internal cues. By the word cues, I mean the signals or the triggers. So the internal cues are triggers or signals coming from the inside of the cell. For example, if certain proteins called sensor proteins inside the cell detect abnormalities in DNA, then they will trigger arrest of the cell cycle. Of course, these sensor proteins, as we said before several times, molecules do not have brains. Molecules are not able to talk, but the language of the molecules is through molecular interaction. So if sensor proteins sense the damage in DNA, then they will bind to inhibitory proteins like the famous P53 protein we're going to focus on today to stop the process of cell division, or to arrest the cell cycle. And why are we arresting the cell cycle in this condition? We are trying to gain time in order to try to correct this damage. But there are also external cues coming from the outside of the cell, like molecular signals that tell the cell about the external environment, and that also affect the decision of the regulatory protein to whether the cell will continue or will be arrested in the cell cycle. But if we consider those factors, whether they are internal factors or external factors, how, as molecules, will they be capable of forcing the cell to arrest or triggering it to continue. This is, again, as I said before, through molecular interactions. It's all about molecular interactions, and it's all about biochemical pathways that are series of chemical reactions that result at the end of the day in a cellular response. So these cues will activate certain signaling pathways. A pathway is a series of biochemical reactions that will result at the end of the pathway in a response from the part of the cell. These signaling pathways will result at the end of the day in activating or inactivating certain core proteins, certain essential proteins. So those core proteins, and we call them core proteins because they are essential proteins, their main function is to move the cycle forward. So principally or essentially, the core proteins are essentially here to activate the cell cycle, to move the cell cycle forward. But if their function is to move the cell cycle forward, how then are we saying that they can stimulate or inhibit the cell cycle? Because regulation is not only about stimulation, but it's stimulation under certain conditions when there is a need for the cell cycle to be stimulated, and inhibition under other conditions when there is a damage or a mutation or an abnormality. Yes, this is very true, but the stimulation and inhibition is not through having two types of core proteins, but it's through having one type of core proteins, one category of core proteins that moves the cycle forward, but we sometimes activate those activators or inactivate those activators, and we call them core cell regulators. So the checkpoints, as we have just said, are regulatory proteins, whether proteins with catalytic enzymatic activity or not. And we also call them core cell cycle regulators. And the adjective or the description core here describes the regulators. So when we say core cell cycle regulators, we mean they are the core regulators of the cell cycle, or they are the essential regulators of the cell cycle. So what are the core cell regulators? They are essential proteins that directly drive the progression of the cell cycle. So essentially, they drive the progression of the cell cycle. They are activators, but how do they activate or inactivate if they are activators? It's by activating or deactivating the regulator itself. So one good example of the core cell cycle regulators are the cyclins and the CDKs, or the cyclin-dependent kinases. The cyclins are proteins that are abundant in the cell, that have a major role in regulation of the cell cycle and progression of the cell cycle. They got their name from the fact that their level in the blood is fluctuating all the time across the cell cycle or along the cell cycle, and we have discussed them in previous videos on the cell cycle. There is a video on the channel talking in detail about the action of the cyclins, cyclin-dependent kinases, and the regulation of the cell cycle. Here, we are just reviewing this in order to start the characteristics of the cancer cells, but we also have the kinases, and these kinases, just like any kinase, phosphorylate their substrate, and they do their action by adding a phosphate group. But since they cannot function without cyclins and their function is dependent on the cyclins, then we call them cyclin-dependent kinases. These cyclins and cyclin-dependent kinases cause key events in the cell cycle to occur. And when I say cause here, I do not mean that they allow these key events to occur. No, they do not just allow, because if you say allow key events to occur, this means that these key events can occur without the presence of the cyclins and cyclin-dependent kinases, and that they are just there to facilitate. No, here we mean by to cause the key events that they essentially initiate those key events or trigger them. So if they are not there, the cyclins and the cyclin-dependent kinases, then the chemical reactions needed for the cell cycle to proceed will not start from the very beginning. What are examples of key events that the cyclins and cyclin-dependent kinases initiate in the cell? The process of replication of DNA replication, and the separation of the chromosomes in the during the process of mitosis using the spindle fibers. But in addition to those key events that are triggered by cyclin and CDKs, they also do something else very important, which is to check that the order of the events in the cell cycle is correct, meaning that G1 should precede S and S should precede G2. And not only this, but also that one phase can trigger the onset of the next phase, meaning that if G1, the termination of G1 triggers, of course, through a series of enzymes and biochemical reactions, the beginning of the S phase. And any disruption that will occur during the cell cycle means what? By disruption here, I don't mean a disruption in the function of the regulators. I mean a disruption in the processes that should be done in every phase. Meaning, I mean that if there is a disruption in G1, there isn't enough nutrition to start. If there is a disruption in S2, that's to say we have an error in replication of DNA, then what will happen is the norm is apoptosis, meaning that the cell will be arrested in G note, and if repair processes fail, then this cell will undergo suicide or programmed cell death. And there is a video that I've uploaded a long time ago on the channel talking about apoptosis. But sometimes we have a disrupted cell with damaged DNA or any other kind of abnormality that skips the checkpoints, that does not undergo apoptosis, although it is an abnormal cell with damaged or disrupted DNA. And in such a case, this cell is an abnormal cell, most probably a mutated cell. However, this cell manages to escape, to bypass the checkpoints of the cell and continues the cell cycle, and therefore is allowed to give daughter cells that will also have mutated DNA, and we will have a mass of abnormal cells that will cause cancer. But cancer is not only about allowing abnormal cells to divide, but it's also about this capacity of the cancer cells to divide very rapidly and to breach and break all the fundamental rules in the cells that are protective. And in this part of the video, we are going to mention some of the characteristics of cancer cells. The first very famous characteristic of cancer cells is that cancer cells do not submit to senescence. What do we mean by senescence? Senescence means getting older. So most cells have a certain limit of cell cycles. We have discussed this before in the video on the telomeres, because you know that every time the cell enters in a cycle of mitosis, and even every time a DNA molecule replicates itself, a part of the telomere is lost. It's true that there is a telomerase enzyme in the cell that tries to rebuild the telomeres that are at the end of the chromosomes and that protect the chromosome. But those telomerase enzymes have a certain capacity. So when it is exceeded, then the telomerase cannot rebuild the telomeres, and the DNA loses its telomere completely and is now exposed to being mutated. And that's why there is a certain, like what we call a clock that counts the number of cell cycles. That clock is the telomere, because every time you do a cell cycle as a cell during mitosis, you lose a part of your telomere. So most of the cells, if you don't remember the details, you can refer to the video on the telomeres and on mutations. So every cell is allowed only maximum 40 to 60 cycles of mitosis, after which it has to be discarded, arrested, to enter permanently in the G node. And this is what we call the Hayflick limit. So if the cancer cells, so the ordinary cells have like an aging process. After 40 cycles of mitosis, for example, the cell will say, I can no more divide. If I divide more than this, I'll have mutant DNA and I'll damage the whole body, and it calls for apoptosis or self-suicide. And you can check the video on apoptosis to understand this. But the cancer cells do not submit to senescence. They can do more hundreds of cell cycles repeatedly in sequence without stopping, and therefore they increase tremendously in numbers. They give huge masses of cells. They outcompete the normal cells that cannot compete with them, and they even replace those healthy cells. Similarly, the cancer cells do not submit to apoptosis. We have just said that any cell with mutant DNA or with lost telomeres, when the telomeres are completely lost, it should undergo apoptosis. It should first be arrested, discarded from the cell cycle, entering the G node permanently, and beginning apoptosis, or this programmed cell death. But the cancer cells do not submit to apoptosis. So apoptosis, as we have just said, is a regulated process of programmed cell death that is targeting the cells with irreparable damage. That's to say, we've explained before that when the cell detects damage, the cell is arrested temporarily in the G node in a trial to repair that damage. But afterward, if the damage proves to be or irreparable, then the cell will undergo apoptosis. That's to say, there will be enzymes that will trigger gradual, programmed, organized, clean degeneration of the cells. Or actually, it's not true degeneration, because it's a very organized process of breakdown of the components of the cell, in which there is recycling of the components of the cell to build new healthy cells. So the cell is prevented from continuing, and it is prevented from getting copies of itself. And this protects the body, as we said before, from cancer. But cancer cells do not obey this. Cancer cells have mutant DNA, and their damage is known to be irreparable. And however, they continue. They do not obey the rules of the checkpoints. Why don't they obey the rules of the checkpoints? Because this same mutation that made it be a cancer cell is the same mutation that allows it not to obey the checkpoints. because if the DNA of the cancer cell is mutated, then the DNA that is expressed into the core regulatory proteins and checkpoints is itself mutated. So the idea here is that the mutation does not only affect the structural DNA in the cell that is related to the process of replication and so on, but it also involves the genes that code for the core regulatory proteins like cyclins, CDKs, and P53 protein. So how can the cell obey apoptosis and obey the checkpoints if the checkpoints themselves are mutated and therefore do not carry out their action properly? That's why cancer cells can skip or pass the checkpoints, and the problem comes from here. You might have been wondering before listening to this or watching this video, how come that we have checkpoints in the cell and that a mutant cell like the cancer cell can continue division? And we have just explained how. So here we are saying it again with more organization. So how can cancer cells escape the checkpoints? The original code for making any protein is stored in DNA. So if you have a code alteration, that's to say if you have a mutation, then this mutation will lead to an altered protein or a mutant protein. And if this mutation, and this happens frequently in cancer cells, occurs in a gene or in a DNA region that codes for a regulator of the cell cycle, then there will be poor regulation or lack of regulation or failure of regulation of the cell cycle. And if the cell cycle is not regulated and is out of control, then we will have mutant abnormal cells that are allowed to divide. And not only this, but also the rate of division itself can become uncontrollable. And these cancer cells, in addition to being mutated, and in addition to have their core proteins, regulator proteins mutated, they also have the proteins that regulate the pace of the cell cycle mutated. And that's why they have an uncontrollable rate of cell division, a very rapid rate of cell division, because the protein that is supposed to stop the mutated cell itself is mutated. As we said before, the cyclins are a group of related proteins that are concerned with stimulating the cell cycle, triggering the continuity of the cell cycle, the progression of the cell cycle. And as you can see in this diagram, we have several types of cyclin. There are cyclins that peak at the junction between growth 1 and synthesis phase. There are other cyclins that peak during the S phase. There are a third group of cyclins that peak during the M phase, and this is normal because we have just said that every one of these is a checkpoint. But here I got the diagram only to show you how the concentration, you can see on the y-axis, how the concentration of cyclin is fluctuant. So for example, before the G1 checkpoint, the cyclin increases in concentration, and once it finishes the check, it is broken down and recycled in the cell. The cyclins, as we said before, regulate the cell cycle. Their concentration fluctuates. They are constantly metabolized, degraded, and reproduced in the cell. And we also said that cyclins are produced to promote. They are not produced to promote or inhibit. They only promote. But how then can they be inhibited? If their molecules themselves become degraded. So cyclins are produced essentially to promote the cell cycle progression, and when they are degraded, they inhibit the cell cycle progression. And as we said before, they are closely related to CDKs. Cyclin dependent kinases, which are enzymes, and the role of the cyclin proteins is to activate those enzymes. And there is a good deal of specificity between the cyclins and the CDKs, which means that every CDK has a specific cyclin to activate it. We also said, we are just recapitulating, that the CDKs are a group of enzymes that are involved with phosphorylation or the direct addition of a phosphate group from ATP. That's why they're called kinases. They phosphorylate their substrates to promote certain cellular activities. Now, cancer cells, as we have said, we are pursuing our discussion of the characteristics of the cancer cells. We began the video by explaining the fundamental knowledge that is necessary to understand the characteristics of cancer cells. And every now and then we leave the main characteristic to discuss a side item about the function of a particular enzyme or to remind you of something related to the cell cycle. Now we have discussed 3 properties of the cancer cells, and the 4th one is uncontrolled cell division. We have just mentioned this, and we are now explaining why. When you have an uncontrolled cell division, it's either because you have too little activity of the inhibitors of the cell cycles or too much activity of the positive regulators. You might be wondering now why I am mentioning inhibitors of the cell cycle. You might want to ask me, haven't you said that the core cell regulators are essentially activators and that the positive or negative regulation comes from their activation or deactivation respectively? Yes, the majority of them are like this, the cyclins and the cyclin-dependent kinases, but there are proteins like the P53 that are essentially inhibitory proteins. So if those inhibitory proteins have very little activity, then the cell cycle is stimulated. Meanwhile, if the stimulators have increased activity, then again, the cell cycle will be stimulated. Those two abnormalities, those are not normal conditions. When you have too little activity of the inhibitors or you have too much activity of the positive cell regulators, this is not a normal condition. And since it is not a normal condition that the inhibitor is inhibited or that the positive activator is activated, this means that these abnormal conditions result from mutations, as we have just said, in the genes that code for the cell cycle regulators. So we might have abnormal forms of the cell cycle regulator. For example, we have just mentioned that the P53 gene is a gene that codes for an important inhibitory protein in the cell called P53. If the gene for that protein, if the gene that encodes that inhibitory protein is itself mutated, then the inhibitory protein will have very little activity or no activity at all, so it will not be capable to arrest the cancer cells and the cancer. And this explains why the cancer cells will be capable of skipping the G1 restriction point of the cell cycle. Another property of the cancer cells, because they have an uncontrollable cell division, is that they can multiply in vitro. And by in vitro, I mean outside a living cell, for example, in a Petri dish or in the lab. They can multiply in vitro without growth factors. The growth factors are proteins that stimulate cell division. So in the cell, in vivo or in the living cells, the normal cells need the presence of this very important protein. It's not growth hormone secreted by the pituitary gland. It's a growth factor that is secreted by every cell. And the growth factor is an endogenous factor, or sometimes coming from other neighboring cells, that triggers the normal cells to divide. When you have the cells in vitro, that's to say outside the living body where there are no cells and there is no growth factor present normally, then you have to add growth factors. So if you have a culture of normal cells and you need them to multiply or divide, then you have to add a growth factor. If you add a growth factor, then the cells will divide. If you don't add a growth factor, then the cells will not divide. But the cancer cells in vitro outside the human body or a living cell can divide and undergo mitosis, whether you add a growth factor or you do not add a growth factor, and that's serious. And that explains why the cancer cells have a great possibility or capability of dividing and dividing and dividing in a very rapid pace to give masses of cells that make the tumor. This is because they do not need a growth factor. But how come that they do not need a growth factor if the growth factor is essential to stimulate mitosis? The answer is simple: because you are not dealing with normal cells, you're dealing with mutant cells. And one of the mutations in the cancer cells allows them to make their own growth factor. So the cell itself makes its own growth factor endogenously, and this growth factor is very effective, and therefore they do not need growth factor from the external environment. Usually, normal cells need growth factor from the external environment, but cancer cells are one example of the cells that, as I've mentioned before, can produce the growth factor endogenously. And this is a very specific characteristic that makes them be able to divide even if they do not receive a growth factor. And not only do they form their own growth factor endogenously, but their growth factor is always stuck in the on position, because we're going to see diagrams at the end of this video or on the next video that show you that the growth factor might sometimes be on or off in the cell. But the cancer cells have their growth factor stuck in the on position, which means that the growth factor is always working. Another third amazing abnormality or incredible abnormality is that the cancer cells do produce their growth factors internally, have their growth factor constantly in the on position, but also trigger their neighboring cells to make growth factors for them. The mutations of the cancer cells also provide them with abnormal properties, like ignoring the signals that try to arrest them from dividing when they are too crowded. And we saw this when we were talking about quorum sensing. You can refer to the video on quorum sensing. Normal cells, when they become too crowded, they stop dividing. Like bacteria, for example, the example that we gave in quorum sensing. But cancer cells, because they are mutant, they do not respond to these signals. The signaling pathway is shut down. All what I've just mentioned tells you that the problem about cancer cells is not only about one mutation, but it's about a group of mutations or accumulated mutations, one after the other, that end up by giving you that abnormal pattern of cells that divide very rapidly, that form masses of cells that do not need any external trigger to divide, that do not obey the instructions of the cells or of the checkpoints. Cancer cells are also characterized by lack of mortality or replicative immortality. We've just mentioned that they do not obey the rules of senescence or aging, and that they can divide even more than 60 cycles. So they are characterized by replicative immortality, meaning that all the cells, after having finished 60 cell cycles of mitosis, are forced to arrest because their telomeres are almost disappeared. And this is what leads them to stop and to undergo apoptosis and commit suicide and die and be recycled into another cells. But when we say that they have a replicative immortality, this means that they continue replication. They do not get old. They do not divide. Because why? Why do they have this ability, although with every cycle of replication, they lose part of their telomere? If you remember, in normal cells, I said it's true that the normal cells have their own telomerase enzyme, but sometimes the rate of division is more than the ability of the telomerase enzyme to rebuild the telomeres. So the problem with the normal cells that makes them die or stop division is that they have a relatively limited amount of telomerase enzyme. But if you know that the mutation, another type of mutation in the cancer cells, I have just mentioned that they have plenty of cumulative mutations, that they can build their own telomerase enzyme in large quantities, and we know that the telomerase enzyme reverses the wearing down of the chromosome tips by building new telomeres after having lost the tips of the chromosomes. Another important characteristic of the cancer cells, which is also due to mutations, is that most normal cells in the body cannot divide unless they're attached to a membrane. Sorry. For example, in the epithelium, the cells that cover the body surface, or the endothelium, the layers of cells that line or coat the body cavities from inside, the cells have a basement membrane on which they rest and divide. If they don't find that resting membrane, most of the time they cannot divide. But cancer cells can divide without these membranes, and that's why they can travel in blood, they can travel in lymph, and they do not only travel in those body liquids, but they're even capable of dividing while they are being transferred in the blood or in the lymph. So they can migrate to new parts of the body, and this migration allows them to make cancer or tumors in new parts of the body, which we call metastases or secondaries. For example, you might have cancer cells in the breast, and then they migrate via lymph and via blood to the bones. So you start having nodules or masses of cancer in the bone that arose primarily from the primary tumor, which was in the breast. Another important thing is that the cancer cells can produce enzymes, and those enzymes can digest the neighboring tissues. And this is why am I saying this here? Because it helps them migrate. So if you have, for example, a hepatic tumor, then it can erode the blood vessels via its own enzymes, digestive enzymes that the cancer cells produce. They digest the liver cells, they digest the blood vessel wall, they reach the blood, and from the blood, they give showers of secondaries everywhere in the body. Because cancer cells divide very rapidly, so cancer cells require a huge supply of oxygen and nutrients and energy. So imagine a huge mass of cancer that is full of thousands of very actively dividing cells with dense nuclei, with dense chromatin. Then they need a lot of energy and a lot of nutrients. And the ordinary blood vessels of the ordinary tissues will not suffice or be sufficient for these cancer cells. So one of their mutations is to have the ability to build their own blood vessels, or what we call angiogenesis. Genesis means formation, and angio means blood vessels, to have a source of oxygen and nutrients. And as we said before, although these cancer cells have all the abnormalities that should trigger the biochemical pathways of apoptosis, yet they do not undergo apoptosis. So here we have a normal cell, and here we have a cancer cell. When a normal cell that is not cancerous has an unfixable or irreparable damage, then it will undergo apoptosis. But if a cancer cell has irreparable apoptosis or damage, it will continue dividing. The same principle applies to the metabolism of the cancer cells. Of course, if the cancer cells are all mutated, most of their genes are mutated, then the genes that control the enzymes and the proteins of the metabolic pathways are themselves mutated, and therefore the metabolic processes in the cancer cells are abnormal. Not only are they abnormal, but those metabolic pathways that are concerned with the production of energy should be very rapid to support the very rapid rate of growth and cell division. So as we said before, cancer cells, they develop a multi-step process in which we have multiple mechanisms of cell division restriction failing before the cancer develops. So when you have a cancerous cell, you have to be sure that since that cell is cancerous and since that cell succeeded in dividing rapidly to form a mass of cancerous cells, then it should have disobeyed or breached hundreds of steps and hundreds of checkpoints in which it did not obey the instructions. So in order to recapitulate all this, we have to say that, or to repeat what we have just said, that cancer cells cannot be cancer cells without having a series of mutations that accumulate in the cell. And these mutations, as we have said before, inactivate the negative regulator. And that makes sense, because if you inactivate a negative regulator, then you are activating the cell. Or on the other hand, the mutation activates a positive regulation. So the first mutation that occurs in the cell, because we say that cancer cells undergo a series of mutations. The first mutation that occurs in a cancer cell is to inactivate a negative regulator. The second mutation is to activate a positive regulator. And the third mutation is the mutation that allows inactivation of a genome stability factor. What is the genome stability factor? The genome stability factor is a gene, and the function of that gene is to be expressed into proteins that prevent the occurrence of mutations or prevent passing those mutations to offspring. Of course, this is a summary of the function. The function is much more complicated than this. For example, the genome stability factor consists of genes that will be expressed into proteins, and these proteins will sense, as I said before, the damage in the DNA by binding with the DNA and sensing the abnormality. They will try to repair this DNA damage, and they will also maintain the telomere cap on the end, because if you maintain the telomere cap, then you prolong the life of the cell and you allow the cell to continue dividing without undergoing senescence. So it's not only about the first mutation or the second mutation or the third mutation, but it's about repetitive accumulations of those mutations and additional mutations, leading to a very large scale mutation in the cancer cell. A very large scale mutation means that inside the nucleus of the cancer cell, you have plenty of mutations that might lead to a serious event in the cell, like, for example, disappearance of a whole chromosome, having an extra chromosome. This is something serious because it's not a gene mutation, but it's more than this. It extends beyond the gene and it might become a chromosomal mutation. And above all this, you have the ability of the cancer cell to escape all the regulatory mechanisms in the cell coming from the outside of the cell, which is also due to mutation. We'll stop here to prevent the video from being very long or too long, and we will have a second part where we will explain the role of P53 in detail. Thank you. If you have liked the video, then click on like, write your comments, subscribe to the channel if you haven't already, and share this video with your colleagues. Thank you, and see you in another video.